Our next speaker is Professor Sam Lee. He is currently Professor of Medicine and Hepatology with research interests in cardiovascular complications of cirrhosis and also viral hepatitis. He has published more than 180 scientific papers in more than 20 books chapters. He is a past president of the Canadian Association of the Study of Liver and International Ascites Club, and he was an editor-in-chief of Liver International between 2008 and 2013. He's from University of Calgary. He's going to talk about cardiovascular complications of cirrhosis and portal hypertension. Sam.
Thank you for introducing me, and thanks for inviting me to come to sunny Riyadh in the middle of Canadian winter. It's when I left Calgary a couple of days ago. I can't remember now. It was a long time ago. It seems like the temperature was -20. So people were complaining that it's cold here in Riyadh. I said, no, this is not cold. Wait till you experience -20, then you might be able to talk. So I've been given this topic, and it's a huge topic, so I want to just present some small sections of it. I have no disclosures to make relevant to this topic. So these are all the different cardiovascular changes in cirrhosis and portal hypertension. And I want to address the first two that I've put in italics: hyperdynamic circulation and decreased effective circulating volume. And over the last 25 or so years, I've done a lot of research in others, including cirrhotic cardiomyopathy, but I'm not going to mention these others. So the cardiovascular system, the body in general, we think is very well designed. So when it becomes abnormal, we have to ask ourselves, what happened? Why isn't it designed so well? So these are some examples of perhaps not so good design. So I know most of you are clinicians, and most of you don't do bench or animal research. So I have to explain why this topic is important, why you should not all immediately go to sleep now, because almost all the rest of my talk is going to talk about my animal research in rats and mice. So the clinical significance of hyperdynamic circulation, where the circulation becomes turned on, increased cardiac output, decreased peripheral resistance and arterial pressure. That's the syndrome of hyperdynamic circulation, and most people agree that it's related, it's involved in the so-called peripheral vasodilation theory of ascites formation. Hyperdynamic circulation and vasodilation in the lungs causes hypoxemia, the so-called hepatopulmonary syndrome, and HDC may worsen variceal bleeding and increases the stress on the heart. So the traditional view that over the past 50 years, most people have been looking at a so-called humoral factor, increased circulating levels of things like glucagon, and actually nitric oxide took most of the attention for the past 25 years, or a peripheral blunted response to vasoconstrictors such as catecholamines. The central nervous system, the brain, actually controls the cardiovascular system. And nobody thought to look at the brain until relatively recently. My lab started doing that about 15 or 18 years ago, because parts of the brainstem and hypothalamus actually control cardiovascular regulation. So this is the nitric oxide hypothesis. Its main proponent has been Roberto Grossman, who's studied this field for 60 years almost. And this is a very important paper published from his lab by Yasuko Iwakiri in American Journal of Physiology about, 15 or 16 years ago. And in knockout mice who have the gene for both eNOS, endothelial nitric oxide synthase, and inducible iNOS, so these rats basically cannot make nitric oxide. Even then, the PVL, the dark bar, portal vein ligation, where you ligate the portal vein, so these rats develop portal hypertension, a prehepatic portal hypertension. When you do that, you can see in the top corners, the mean arterial pressure is about the same, but even in the knockouts, you knock out nitric oxide, cardiac index, and systemic vascular resistance, on the right side of the two bar graphs, are still abnormal, suggesting that it's not just nitric oxide, something else is causing hyperdynamic circulation. So our hypothesis is that central neural activation causes the hyperdynamic circulation. So I have to show you some neural physiology, which you probably never learned or forgot instantly when you learned this in medical school. But there's a marker called CFOS, which is known as an immediate early gene. And when neurons in the CNS are activated, CFOS lights up. CFOS is activated in those neurons, and it makes FOS. And we can stain for FOS by immunohistochemistry. So it's well accepted as a marker of neural activation. When neurons get activated by whatever stimulus, we can look and find FOS, okay? And you know about the nucleus of the solitary tract. It's a primary relay station. It's one of the key cardiovascular relay centers, but there are several others. So these other ones include so-called ventrolateral medulla, the paraventricular nucleus, and the supraoptic nucleus. These are all cardiovascular regulatory centers in the brainstem and hypothalamus. So this is a study we did in portal vein stenosis, and we did the operation where we stenose the portal vein. So these rats develop portal hypertension. And even day one after the surgery, so you'll see the sham control. So these rats have just had the laparotomy, but no ligature of the portal vein. PVS, portal vein stenosis. And what you see here is the number of FOS neurons. So neural activation in all these centers, and paraventricular nucleus, supraoptic nucleus, nucleus of the solitary tract, and ventrolateral medulla. Even day one, they all light up, and it happens, keep measuring day 3, day 5, day 10, when the hyperdynamic circulation is well established between day 5 and 10. But even before then, these neural regions light up. So what does that mean? Here's the actual immunohistochemical detection. So you'll see here the left side is a sham control of the nucleus of the solitary tract, and on the right side, panel B, is you can see these black dots are FOS-reactive neurons in the NTS region. And in the bottom panel, C and D, the sham control, and on the panel D is the portal stenosis. So B and D are the portal hypertensive rats. A and C are the controls. And you see D, the supraoptic nucleus, really lights up. So those neurons, those black dots, are all concentrated in the supraoptic nucleus, and in B, the solitary tract. And the immediate question was, what kind of neurons are these lighting up? And the obvious answer, we thought, was they're catecholamine-containing neurons, i.e. sympathetic nervous system neurons. And in fact, that turned out not to be. So here's a double stain where the tyrosine hydroxylase, which is a marker of catecholamine neurons, are green, and FOS positive are red. And you see in the nucleus, in the catecholamine in the cytoplasm. And you see, they don't overlap. There's maybe a couple of neurons where they overlap, but by and large, FOS reactivity is in a different neuron. And rather, there are dozens of different types of neurons in the CNS, and we thought, well, this is kind of fruitless. We don't want to keep trying to find out, stain for every neuron, trying to find out what it is. Let's try to determine why they're lighting up, what's causing the neural activation. And in order to do, well, here's the sequence of cardiovascular changes. So neural activation precedes the development of hyperdynamic circulation. It's probably somehow causing the hyperdynamic circulation. So one of my graduate students, long time ago, very clever Chinese fellow, Daishen Song, his PhD project with me was he actually did a micro-injection of antisense. So you can block CFOS activity by injecting antisense oligonucleotides to CFOS into the brainstem, in this case into the NTS of rats. And you can imagine this is like super delicate. It was a 26 or 27 gauge needle that he used, and there's a stereotaxic apparatus you can use to accurately inject antisense into these into the NTS. So we're trying to block CFOS activity. When you do that, so panel A shows this. A and C are sham controls. We've in the panel C down below, bottom left, we've injected antisense to CFOS. B and D, we've injected the sham and the portal vein stenosis. And you see in the top right, the portal vein stenosis that got the sense. So sense is a control. So it won't affect, it will not block CFOS. So in that, you can see that region just to the right and left of the CC central canal, the nucleus of the solitary tract. There's FOS lighting up, as I showed you before, with portal vein stenosis. And when you inject antisense in the bottom panel, the bottom right, panel D, you see that the FOS is all gone. It's disappeared. So it shows that the antisense worked. And then we looked at the cardiovascular effects of that, and you can see that in the panel, far right panel, PVS-AS, that's the portal hypertensive rat that got antisense, so CFOS activity is blocked. That actually makes the circulation go back to normal values. So it blocks the hyperdynamic circulation from developing. So what this means is if you block neural activation, they do not get hyperdynamic circulation. And then the question is, so obviously that part of the CNS is activated. What's activating it? And our hypothesis was that somehow a signal, because the brain does not just turn on the circulation for no obvious reason. It must be reacting to something, some signal, and we thought the signal is from the gut that has portal hypertension, mesenteric congestion, and that is relaying a signal by the vagal afferent nerves to the CNS saying that I'm ischemic or I'm congested, turn on the circulation, get that blood flow moving to relieve the ischemia or the congestion. So in this series of experiments, we looked at, we actually measured the electrical neural activity of the vagus nerve. And if you tie a portal vein occluder, a balloon, inflatable balloon around the portal vein and inflate it, it causes acute portal hypertension. You see the number of the spikes, you can count the neural spikes, and there's a dramatic increase in vagal neural activity. And capsaicin is a neurotoxin that if you administer it, it blocks vagal afferent nerves. So it's a selective afferent denervation. And when you do this, the PVS rats that the far right black solid graph, you'll see that the number of FOS reactions, if you get capsaicin painted on this is a control nerve in the gut versus the V is the vagal afferent. So even though they did not reduce completely to normal, treating the vagal afferents with capsaicin had a major effect on FOS reactivity. So it blocked partially the central stimulation. And vagal capsaicin treatment also reduced the hyperdynamic circulation. So both bars, on the right, the right two bars, the solid bar and the cross stripe bar, show that treating these with capsaicin does partially, not entirely, but partially reduce the hyperdynamic circulation. So the summary of brainstem hypothalamic neuroactivation leads to hyperdynamic circulation. So the regulation is central. It's not peripheral. It's not at the blood vessel level. It is centrally mediated. The gut signal, we think, is probably mesenteric congestion carried by vagal afferent nerves. So this is Zakim Boyer's textbook that Sungu Beck and I co-wrote this chapter on cardiovascular changes in portal hypertension. And this is the diagram from this that it's a somewhat complicated schematic, but you'll see at the bottom in the gut, the bottom part of that schematic, mesenteric venous congestion and blood pooling lead to some kind of signal carried by vagal afferents to the CNS that turns on the circulation. I'm getting a little bit short on time. This is why study of hydrodynamics is important. I don't know how this picture was taken at the exact perfect time, but you can see here that somehow hydrodynamics, now you don't drink beer in this country, so you probably don't appreciate this, but if you clink 2 beer glasses for a cheers or something very hard, this is the kind of bad thing that can happen. So because I'm running a bit short on time, I'm going to whip through this very quickly because I realize this is going to so where does the extra blood volume, we know 30-40% cirrhosis, portal hypertension, blood volume increases, where does it go? Basically, it pools in the splanchnic and mesenteric veins. So this is a bunch of experiments we did about 10 years ago showing that. And in the interest of time, I'm not going to go over these in detail. But I'm happy to make this presentation available. Anyone, you just have to email me, I'll send you this talk. So you can look at this yourselves in detail. And bile duct ligated, so these are cirrhotic rats now. They have cirrhosis due to bile duct ligation. Their gut veins show less compliance, so you increase the pressure and you draw this pressure volume slope. The slope of these lines, one is a sham operated control, one is a cirrhotic, and they're significantly different. And if you remove neural influence by tetrodotoxin, which takes out all neural influence, it's a sodium channel poison. So the black lines with their black circles and squares are after tetrodotoxin. It restores the slope to normal, to the same as the sham control.
2 minutes.
And, okay, sorry, I'll just end up with this. This is an experiment that we published many years ago showing that the BDL has impaired venoconstriction. It doesn't squeeze the gut veins to defend the circulation after hemorrhage. And If you block that response with the selective nNOS inhibitor, SMTC, it restores, it makes that go back to normal. So what this means is that my final slide is there is central neural dysregulation. It's a peripheral to central signal via vagal afferents. The decreased effective circulating volume is due in part to cardiac dysfunction, but also gut venous pooling. And nitric oxide from nNOS is probably the peripheral effector. I'm sorry I put you through this very sort of complicated animal pathophysiology talk, but I hope you learned a little bit about pathogenesis of hyperdynamic circulation and cirrhosis. Thanks for your attention.